Hello, doctor and my fellow friends. Today, I'll be the representative of my group, which is Group 6, SEN1A. My name is Nurasha binti Mamazaki, and our topic for video representation is mitochondrial bioenergetic and metabolism, implication of human health and disease. Next, this is our group member list. Shawal will talk about history. Farhan, Fahim, and Faris will talk about discussion. Narin will talk about advantages. I, as Nourisha, will talk about disadvantages, while Shaira and Sari Karnisa will talk about application in daily life.
The study of mitochondria and their role in bioenergetics and metabolisms dates back to the early 20th century. Richard Altman discovered granules within cells in the 1890s and termed them bioblasts, which were eventually recognized as mitochondria. However, it wasn't until the mid-20th century that the actual importance of the mitochondria in cellular energetics became clear. Otto Warburg made groundbreaking discoveries about the relationship between mitochondria and cellular respiration in the 1930s. He discovered that cancer cells, even in the presence of oxygen, rely heavily on glycolysis, a phenomenon now known as the Warburg effect. This led to the discovery that mitochondrial malfunctions and altered metabolism could be linked to a variety of disorders. The development of key experimental approaches increased the understandings of the mitochondrial bioenergetics. Albert Leininger developed a way to isolate intact mitochondria in the 1940s, allowing researchers to explore their structures and functions. The advance of polarography and the Clark electrode in the 1960s and 1970s permitted the monitoring of oxygen consumptions by mitochondria, revealing insights into mitochondrial respirations and ATP synthesis. Peter Michel's discovery of the electron transport chain and the chemiosmotic theory in the 1980s laid the groundwork for understanding mitochondrial bioenergetics. Michel's research demonstrated how electron transfer and proton pumping across the mitochondrial inner membrane produce a proton gradient, which is then utilized by ATP synthase to generate ATP. The field of study was further improved by advances in molecular biology techniques. The discovery and sequencing of mitochondrial DNA, mtDNA, in the 1980s enabled researchers to investigate mitochondrial genetics and their functions in metabolic illness. The concepts of mitochondrial illness, which are caused predominantly by mutation in the mtDNA or nuclear genes involved in mitochondrial functions, gained popularity. Today, research in mitochondrial bioenergetics and metabolism is helping us better understand human health and illness. High-resolution respirometry, metabolomics, and proteomics, for example, allow for truth investigations of mitochondrial activity and metabolic pathways. The mitochondrial malfunctions has been linked to a number of diseases, including neurological disorders, metabolic syndromes, cancer, and aging-related disease.
Next slide is discussions. Mitochondria is very important in bioenergetics and metabolism, and their dysfunction can cause danger to human health conditions. Next is energy production. Due to their ability to produce adenosine triphosphates or ATP, the main source of energy for cells through oxidative phosphorylation, mitochondria are frequently referred to as the powerhouse of the cell. Next is metabolism regulation. The Krebs cycle and the citric acid cycle are two of the metabolic routes in which mitochondria participate. They break down proteins, lipids, and carbs to create ATP and other compounds required for different cellular functions.
Next is mitochondrial DNA mutations. Mutations in mitochondrial DNA, mtDNA, can cause mitochondrial diseases. These genetic changes affect ATP production, the electron transport chain, and mitochondrial protein synthesis. Next is apoptosis regulation. Mitochondria also play a crucial role in the regulation of programmed cell death, known as apoptosis.
The very first advantage of mitochondria in terms of implication, human and disease, are metabolic regulations. Mitochondria play a vital role in regulating metabolic pathways. It also acts as metabolic sensors. It adjusts their activity based on nutrient availability and energy demands, ensuring proper metabolic balance. It is also essential for maintaining metabolic homeostasis and preventing metabolic disorders like diabetes, obesity, and metabolic syndrome. Next advantage are mitochondria act as an energy production. Mitochondria generate the majority of cellular energy in the form of adenosine triphosphate ATP through oxidative phosphorylation. It is also crucial for sustaining various cellular processes, including muscle contraction, brain function, and organ maintenance. Last but not least, mitochondria is a cell signaling and apoptosis. Mitochondria participate in cell signaling pathways, and the regulation of programmed cell death is known as apoptosis. They release signaling molecules and contribute to the activation of apoptotic pathways. It is also essential for maintaining tissue homeostasis and also eliminating damaged cells.
For the disadvantages, I found several disadvantages for this method. First, energy deficiency. Mitochondria are responsible for generating adenosine triphosphate or ATP, the primary energy currency of cell. Any impairment in mitochondrial bioenergetics can lead to an energy deficiency, resulting in reduced cellular function. This can affect various organs and tissues, leading to symptoms such as fatigue, muscle weakness, and exercise tolerance. The second disadvantages are oxidative stress. Mitochondria are also a major source of a reactive oxygen species, ROS production. ROS, when produced in excess, can cause oxidative stress, which can damage cellular components such as DNA, proteins, and lipids. Prolonged oxidative stress can contribute to the development of various diseases, including neurodegenerative disorders, cardiovascular disease, and cancer. The third disadvantages for this method was mitochondrial DNA mutations. Mitochondria have their own separate DNA known as mitochondrial DNA, mtDNA, which is vulnerable to mutations. Mutations in mtDNA can impair mitochondrial function, leading to energy deficiency and increased oxidative stress. These encephalomyopathy, lactic acidosis, and stroke-like episodes called Miller syndrome are associated with specific MT mutations. Next, metabolic disorders. Mitochondria are involved in various metabolic pathways, including glucose and fatty acid metabolism. Disruptions in these pathways can lead to metabolic disorders such as diabetes, obesity, and metabolic syndrome. Impaired mitochondrial metabolism can result in reduced glucose utilization, insulin resistance, and dysregulation of lipid metabolism. Final disadvantages are age-related decline. Mitochondrial function tends to decline with age, leading to reduced energy productions and increased oxidative stress. This age-related decline in mitochondrial function has been implicated in the aging process and age-related diseases. Mitochondrial dysfunction can contribute to age-related conditions such as neurodegenerative diseases, cardiovascular diseases, and sarcopenia, which is loss of muscle mass and strength.
Application in daily life. First is DNA testing. Trace geographic distribution of genetic variation for the investigation of expansions, migrations, and other pattern of gene flow in DNA. For example, analyze a variety of forensic samples like old bones, teeth, and hair that we use to testing DNA. Second is in forensic terms. Mitochondrial DNA were maternally inherited, so it can allow the forensic scientist obtain information from old items of evidence, especially with the cold case and small pieces evidence that contain biological material in forensic investigation.
Other application of mitochondria in daily life They were also used by athletes. It because mitochondria contain oxygen and carbohydrate like sugar that will cause substrate. It can produce the energy by make your heart beat and your muscle perform when they consume it. Lastly, mitochondria were also used in respiration term. It because it needs oxygen to convert the energy stored in the macronutrient to adenosine triphosphate or ATP as the universal energy in the cell. The enzymes of the respiratory chain are embedded in the inner mitochondrial membrane.
Okay, that's all. Thank you for your splendid time for listening our presentation. Bye.